Hi students, this is our next lecture in the Biology 2 online course. In this lecture it will be really short. We will discuss blood. Blood is essentially a very nicely designed transportation system. It's a fluid, liquid that flows through the blood vessels. Uh, and it contains a lot of different things. It contains all kinds of organic and inorganic molecules and several types of cells, cellular components. This picture shows you human blood, which was collected into a tube, and this tube was either just let stand on a bench for about 1 hour or so, or was centrifuged, spun in the centrifuge. And when we treat the blood like this, we see that the blood separates into plasma and cellular components. Plasma is liquid. In this picture, it's the top part. It's lighter colored. It's your textbook mentions the term straw colored liquid, and it is largely water. You see, 91% of the blood plasma is water, but it also contains proteins like albumin, albumins better in plural, globulins, and fibrinogen, and a variety of other nutrients like amino acids, sugars, lipids, uh, complex simple carbohydrates, fatty acids, etc., etc. It also contains hormones. We will talk about hormones in more detail in 2 lectures from now. Then it contains electrolytes, sodium, potassium, calcium, etc. So a variety of ions which are important for functioning of the organism. Then cellular components, in this picture they are represented by what we call buffy coat. It's a narrow whitish stripe which contains white blood cells and platelets, and a wider uh, piece which is red blood cells, packed red blood cells. These red blood cells are the most numerous cellular components. There is approximately 5 million of them in each cubic millimeter of blood. It's a huge number. On the other hand, white blood cells are 7 to 9,000 per cubic millimeter, and platelets about 250, uh, maybe a bit more than that, 300,000, 400,000 per cubic millimeter. Uh, Now, let's have a bit closer look at cellular elements of the blood. Uh, you see a section of a blood vessel here and a number of cellular elements kind of popping out of it. Red blood cells, what is their primary function? They carry oxygen throughout the body. We'll later see that this oxygen is reversibly attached to hemoglobin and carries by the blood fed to the cells of the body. Then white blood cells, they are shown as white cells with white cytoplasm and bluish nucleus. They are their primary function is to help fight infections. Then there are platelets shown as little uh, yellow-colored circles, really small ones. Their function is to help control bleeding. So they are mechanically occluding damage in blood vessels, and also they produce clotting factors which stimulate blood clotting. Now, each of these cellular elements, a little bit more about them in detail. Red blood cells. Uh, they're small. They have a shape of a disc. Uh, as you see in upper left corner, they don't grow, they don't divide, and they don't make any new proteins. Uh, why is that? They mature in bone marrow from precursors called stem cells. Stem cells have nuclei, like all eukaryotic cells, right? But then at some point of their maturation, they kick out their nuclei. See, they are very highly specialized cells. They don't need to divide, so they don't need DNA. They don't need to synthesize any new proteins, so they don't need DNA or RNA for that matter. Uh, what is their function? They carry hemoglobin. Uh, they are packed with molecules of hemoglobin. What is hemoglobin? It is shown on the right part of this picture. It's a protein which consists of four large chains of amino acids. Uh, each chain, you see, they are shown in golden color, in like threads, curved, coiled threads inside red blood cell. Uh, each of them has a non-protein part called heme. It's a chemical group which uh, does not consist of amino acids like proteins, but it has a different chemistry. What you need to know is that each heme necessarily contains iron. Iron. Synthesis of heme, synthesis of hemoglobin without iron is impossible. And iron is not a major component of our diet, and iron cannot be produced in the body. So there are only uh, certain foods that contain iron, and we need to eat them to have a well developed, full-blown synthesis of hemoglobin. Hemoglobin. Then to the right, you see hemoglobin molecules separately from red blood cell, and in the right lower corner, you see hemoglobin molecule where each heme is combined with something that is shown blue. This something is oxygen, actually. So each molecule of hemoglobin carries four molecules of oxygen, one per each heme. That's the most important function of hemoglobin. This oxygen is what is delivered to our cells, what makes them alive, what makes them able to synthesize ATP in sufficient numbers. So again, function of hemoglobin. Hemoglobin has the ability to bind oxygen reversibly and to carry it to tissues. Basically same picture as in the previous slide, but somewhat uh, bigger scale. You see hemes that are combined with oxygen molecules, and in this picture, oxygen molecules kind of dominate over hemes. So you don't even see a heme which is not oxygen. What does it mean reversibly? It means that there is a certain strength of binding between oxygen and hemoglobin. It can be bigger, it may be smaller, but it's never as tight as in uh, molecules that have chemical bonds. Like the strength of binding of oxygen to heme is much, much less than the strength of binding of oxygen to hydrogen in water molecules. And so under certain conditions, uh, a heme can attach oxygen, and at some other conditions, oxygen can detach from heme. Uh, Also, another function of hemoglobin, it facilitates the exchange of carbon dioxide between the lungs and tissues. And so in the uh, uh, in the lung, remember where the uh, amount of carbon dioxide in the blood capillaries is much higher than the amount of uh, carbon dioxide in the alveoli, oxygen diffuses from blood into the alveoli. So carbon dioxide diffuses from blood to alveoli. To be able to diffuse, it must first detach from the hemoglobin. And so that's another function of hemoglobin. So hemoglobin functions as the primary medium of exchange of oxygen and carbon dioxide. Basically, again, so to summarize previous lectures and to kind of make a synthesis with this lecture, in the lungs, oxygen is high in the alveoli, low in the blood capillaries. So it diffuses from the alveoli into the red blood cells. There it binds to hemoglobin. Then this newly changed hemoglobin, called oxyhemoglobin, travels in the circulatory system, reaches the periphery, reaches cells of the entire body, the skin, the internal organs, the muscles, bones, etc. There, the concentration of oxygen in the blood is higher than in the tissues, so the oxygen diffuses from uh, the blood, uh, from the red blood cells into tissues. Before it diffuses, it needs to detach from hemoglobin. Uh, Carbon dioxide, exactly the opposite. In the lung, uh, right, in the lung, carbon dioxide is high in the blood capillaries, low in the alveoli. So carbon dioxide diffuses from the red blood cells into alveoli. Before it diffuses, it needs to detach from hemoglobin. And then in the tissues where cells produce carbon dioxide all the time, and the blood is enriched in oxygen and poor in carbon dioxide. Again, carbon dioxide diffuses back into red blood cells, right, and attaches to hemoglobin, forming what we call carboxyhemoglobin. So that's the function of hemoglobin. Medium of exchange of oxygen and carbon dioxide in the body. Uh, Now, a bit about white blood cells. On the left, you see this microscopic picture where red blood cells are shown in pale red or pink, and white blood cells are, they look like blue spots. Actually, they are colorless, but they have large nuclei which stain blue. Right? So these blobs with blue coloration, they are white blood cells. Note that the shape of the nucleus can vary from one white blood cell to another. It may be rounded, it may be like a rod, it may be like several little rods, segmented nucleus, etc. White blood cells are also called leukocytes. Right? And like as we said, their major function is to protect us from infections. There are several types of them in this little chart or table. You see drawings of different types of white blood cells and a short summary of their lifespan and function. Uh, Lifespan is pretty short, except the lower two categories. We will talk more about that in the lecture about the immune system. Uh, the function of these white blood cells are always aimed at defending us from infections, but they are kind of, there's a division of labor between different kinds of white blood cells. The most numerous kind is the so-called neutrophil. It's this sort of your typical generic white blood cell. And, it has either rod-shaped or segmented nucleus, never rounded, never rounded. It basically what it does, how it defends against infections. Some neutrophils are able to phagocytosis. Remember, we mentioned phagocytosis in lectures about protists. We said that amoebas feed by surrounding their prey with pseudopodia and engulfing it. This is also what some neutrophils do. They internalize bacteria. They internalize some pieces of foreign tissue, etc. Literally eat them. They spray them with digestive enzymes and destroy them. And also many kinds of neutrophils are able to produce chemicals which stimulate inflammation, inflammatory response. Then second from top, eosinophils, beautiful cells. They resemble this kind of like Japanese flag, rising sun. They specialize in defense against parasites, especially helminths, those worms, flat worms, round worms, segmented worms. They are able to produce special kinds of chemicals which not just stimulate inflammatory responses like in neutrophils, but stimulates more targeted responses against parasites. Third from below, basophil. It's a lymphocyte which usually, conventional methods of staining, it stains dark blue. It has a highly segmented nucleus, and it is responsible for inflammatory response just like neutrophils. Plus, also, basophils participate in allergic reactions. Then fourth from right or third from, or fourth from top or third from bottom, monocytes. These are cells which do not really function in the blood. To function, they need to leave blood vessels, and when they leave blood vessels, they morph into, we say, macrophages, cells that are able to phagocytose, like amoebas, like some neutrophils, but they are larger. And their function is participate in immune reactions, immune surveillance, detecting foreign invaders and eating them up, digesting them with digestive enzymes. And then lymphocytes, first from bottom, second from bottom. There are two kinds of them. They both participate in specific immunity. The so-called B lymphocytes produce antibodies, special protein molecules that fight foreign invaders. And T lymphocytes participate in cellular immune responses. I will not talk much about this now. We will have a special lecture about, next lecture will be actually about the immune system. We will recall lymphocytes and talk more about their functions. And to the right, you see scanning electron microscopy pictures of white blood cells. And they look beautiful under the scanning electron microscope. They're significantly larger than red blood cells, not to mention platelets. And their surface contains thousands over thousands of bulging molecules called receptors. Some of them participate in recognition of foreign agents, others participate in adhesion of white blood cells to walls of blood vessels or to tissues. Okay, now we did not, these slides do not include a special discussion about platelets, but I'll jump to function right away. The major function of platelets is control bleeding, right? And there are two ways platelets do it. First of all, what are platelets? They are very tiny uh, pieces of cells. They're not cells themselves, but there are special cells in bone marrow. You might remember their name, or I don't insist that you remember their name. They're called megakaryocytes. And these special cells break, shatter in gazillion of little pieces. Then these little pieces exit bone marrow and circulate in blood. They are platelets. When your blood vessels are damaged by cut or impact or something, and then the bleeding begins. But blood vessels constrict, right, reducing the blood flow, and then these, you see, third picture from top, platelets, tiny little pieces of cells, they aggregate, accumulate in the site of damage. They become sticky, they clump together and form platelet plug. So they're like a mechanical stopper that to some extent it helps to stop bleeding, not entirely. And then what happens is blood clotting. Blood clotting is basically it is forming of a protein called fibrin. Fibrin is a non-soluble protein. Most proteins dissolve in water pretty easily, but fibrin does not. So it remains solid. It does not dissolve in blood. And it creates a mesh that it surrounds the platelets. It grows. It stops the regional or slows down the regional blood flow, and normally it completely stops bleeding, unless of course it's not a major damage to especially arteries. On the right, you see a mechanism of blood clotting, and I have to tell you that it's a pretty complex mechanism. We study it in more detail in human anatomy and physiology, and especially in pathophysiology class. But I just urge you guys to look first at the bottom of this chart, and it says soluble fibrinogen in plasma. Then there is a blue arrow, and it becomes the soluble fibrinogen becomes soluble fibrin. For that to happen, and for the blood to clot, right? Clotting of the blood is the same as forming of the insoluble fibrin. You need to have a special protein which acts as an enzyme. This catalyst of this conversion of fibrinogen into fibrin. It is called thrombin. And thrombin, as you can imagine, thrombin does not exist in its active form in the blood, because otherwise all our blood would just clot and not move. But it becomes active when there is damage to blood vessels. In the blood there is an inactive precursor of thrombin called prothrombin. And in the presence of special factors produced by platelets and also by damaged blood vessel walls, it becomes thrombin, and then it controls or catalyzes the conversion of fibrinogen into fibrin. You need to everyone needs to have some idea about thrombin and prothrombin, because we use this jargon terms like our blood is maybe thinner, maybe thicker, right? And there are blood thinners, like aspirin is a blood thinner, or heparin is a blood thinner, Coumadin is a blood thinner, but it's it's lay jargon terms. What actually happens is that blood can clot faster or slower. When it when it clots faster, we say that it is thick. Right? When it uh, clots slower, very slow, or not at all, we say that it is thin. You probably heard about the condition when the blood does not clot at all. It's called hemophilia. Right? Uh, and there is a number of conditions when blood clots too fast, and when we say it's thick and it needs blood thinners, and to know exactly when the blood is thick, when it is thin, when we need blood thinners, when we need to stop blood thinners because the blood is too thin already. For that purpose, doctors, laboratory people in clinical laboratories measure the so-called prothrombin time, also known as pro time. It's the time that uh, has to be taken for prothrombin to become thrombin and to form and to catalyze the forming of fibrin. Uh, so the higher is prothrombin time, the thinner is the blood, because it takes more time for prothrombin to become thrombin and to activate blood clotting. The smaller is the prothrombin time, the thicker is the blood. And let me tell you this, when I was young, many years, ages ago, when the Earth crust was too hot, so people were still jumping up and down all the time. Uh, Back then, in the 1970s, 80s, our teachers at medical universities and Kiev Medical Institute, where I was when I was a student, they emphasized more on conditions when the blood does not clot or clots too slowly. Too slow, the blood clotting is too slow. Hemophilia and other deficiencies of clotting factors. But nowadays, well, hemophilia still exists, of course, but it is pretty noticeable and it can be treated regularly, and the people that have hemophilia are under attention of medical doctors. So they are more or less okay. Of course, it's a dangerous condition, but they receive medical attention. And nowadays, people whose blood clots too fast, they are the major group of risk. These people can develop blood clots at any time if they don't take blood thinners. And these blood clots can travel to the brain, causing stroke, or to the pulmonary artery, causing pulmonary embolism. I already told you guys that I'm one of those lucky people who had uh, pulmonary embolism and survived, and actually now my blood clotting is pretty much under control. So remember about this, and remember, I urge you to remember and to understand these two terms, prothrombin and thrombin, and what is their role in blood clotting. One more uh, problem which is very prevalent, widespread problem, anemia. So many people have anemia. In the United States, uh, every sixth person has signs of iron deficient anemia. It's a lot of people. They may feel more or less okay, but they are under risk because anemias tend to worsen with time if not treated. What is anemia? Generally, anemia is defined as insufficient amount of hemoglobin. And there are two major sorts of anemia. One is because there is a low number of red blood cells. And the other is when the number of red blood cells is normal. but each red blood cell does not have enough hemoglobin. And it may be combined. It may be low numbers of red blood cells and low amount of hemoglobin in each red blood cell. Now, why iron is so important? Why do so many people have iron deficient anemia? Because if you do not have enough iron, your cells cannot synthesize hemoglobin. The precursors of red blood cells in bone marrow have to synthesize all hemoglobin that a red blood cell will ever have. A mature red blood cell does not synthesize any new proteins. And if there is no iron, then there is no heme. If there is no heme, there is no hemoglobin molecule. So please know this, and I must say that students, of course, they are not the richest people in the world, and they try to subsist on foods that are cheaper, and sometimes they just forget about the need to get iron. You don't get much iron if you eat ramen or something. It may make you less hungry, but it will not provide you with enough iron. So I understand that you cannot buy plenty of fresh fruit and vegetables every day, but still do not completely neglect foods like dry apricots, berries, watermelons, legumes, spinach, etc. They do have a lot of iron, and uh, you suffer if you don't get enough iron. Okay, I believe this is the end of this really short lecture. I tried to make it as close as possible to your textbook. The entire material in your textbook is in just on two pages, section 11.11, and next time we will proceed to the discussion about the immune system. Okay, best wishes. See you next time.